Okay, thank you, Andre. And so I will shift gears a little bit and talk about the adaptive immune system and how it is shaped by the microbiota. And I will give a little introduction to microbiota. So as all of you know, we are colonized by a lot of bacteria which are expressing a lot of genes, so a lot more than expressed by the human cells. And of course, the microbiota plays a lot of important functions. Yeah? So we really have problems when we don't have any intestinal microbiota. So they, for example, help us to digest complex carbohydrates, and they also protect us against pathogens by providing colonization resistance, so occupying niches inside our intestine or on the skin or on other barrier organs so that pathogenic bacteria cannot settle there. And of course, an important function that is the education of the immune system. And as you see here in this picture, actually there's very little which separates the bacteria which are in our intestine from the immune system. So there's really only a single cell layer, the epithelial cell layer, and then the mucus that they secrete, and antibodies, the IgA, which somehow constitute part of the intestinal barrier and separates the bacteria from the immune system. But of course you can imagine that there's a lot of crosstalk between the microbiota and the underlying immune system in the gut or any other barrier tissue. So already there's been a lot of findings where changes in the microbiota have been associated with disease. So already Hippocrates formulated that actually all diseases begin in the gut, and he thought that okay, you need the right nutrition to stay healthy and so on. But there have been recent and for a long time a lot of findings that chronic inflammation is actually associated with a dysregulated gut microbiome, and but we don't know whether that is really a cause or consequence of the inflammation. But there have been already some pathogenic links identified between certain bacterial species and autoimmunity, and has also been shown that germ-free mice, for example, are resistant to inflammatory bowel, induction of inflammatory bowel disease. And there have also been a few findings that, in particular, microbial products, so metabolites from bacteria, can actually directly regulate, for example, the homeostasis of regulatory T cells. And here are some prominent examples of the recent years. So one bacterial group which probably all of you have heard of, the segmented filamentous bacteria, have been shown to be required for a lot of different autoimmune diseases, such as arthritis, colitis, and MS in mice. But they also confer some protective function as they instruct the immune system, are important to instruct the immune system to be effective against Cytobacter infection. So a mouse model for enterocolytic colitis. And then, of course, there's also been a study in the group of Jane Danska, where they've actually shown that the microbiome can actually control the prevalence or the susceptibility to get autoimmunity. So as you know, many autoimmune diseases there's high susceptibility for females, and in using the diabetes mouse model, they could actually show that this sexual prevalence for diabetes in this model is actually controlled by the microbiota. So it's only logical, so that if dysbiosis is somehow associated with chronic inflammation, whether we could somehow treat chronic inflammation by restoring the microbiota. And there have been actually several trials now where they've tried to treat ulcerative colitis by giving them the fecal microbiota of a healthy person. Yeah, so there have been some placebo-controlled random trials, and there are actually two trials here which were published in back-to-back in Gastroenterology in 2015, where they had very contrasting results. Yeah, in the one trial, they found, although it was of very low sickness, they found that they could reach clinical remission in ulcerative colitis patients which received fecal microbiota of a healthy donor. In a trial which was done at the same time, they actually found no significant difference between the patients who received the microbiota and who did not receive, or who received autologous microbiota, their own microbiota, again, or placebo. And then very recent, there was another trial where they treated patients with fecal microbiota transplantation, and here again they saw a significant benefit for patients who received it. So there's very contrasting results, and what they all these authors, what they speculated that's really important, what is the donor. Yeah? So there are some donors whose microbiota is, let's say, more beneficial than others. So it's not really clear what is actually protecting or the therapeutic effect of the fecal microbiota transplantation. So the question we ask actually, can we identify, or are there actually bacteria which can drive or protect us from chronic inflammation? And could this be something that we could maybe somehow improve the fecal microbiota transplantation, or to make it more selective? And a model that we use to study this is the T-cell transfer-induced colitis model. It's a model where you take naive T cells and you transfer them into RAC-deficient mice. And the naive T cells, they will homeostatically proliferate and they will start reacting against the intestinal microbiota. Yeah, in the absence of any regulatory T cells, in the absence of immune regulation. And in the course of 2 to 3 weeks, these mice then will develop a wasting disease and inflammatory and colitis, so inflammation of the gut. And this inflammation can be quite drastic. As you see, if you look at a healthy intestine, so it's, you see nicely formed stool pellets. And when you look in histology, you see a nice structured architecture. You see all these white kind of cells. These are goblet cells, so the cells which are secreting the mucus and are important for maintenance of the intestinal barrier. In the colitic mice, you see that the intestine is thickened and shortened, and you don't see any more stool pellets because the mouse has diarrhea. Sorry, and if you look in the histology, you see a complete disappearance of all these goblet cells, so there's no more of these white cells. You see huge mononuclear infiltration, so these mice are really sick and the intestine is really inflamed. So Jakob, a PhD student who is also now doing a postdoc in the group of Anna McPherson and Brand, has actually used this model to characterize the immune response in these. So what he did is he induced the colitis and then re-isolated the T cells from this colitis and characterized them. And looked, here in this example, looked for cytokine expression. And what you could see is in all the organs that he looked at, in the colon, small intestine, spleen, and lymph node, that most of the cells are actually expressing interferon gamma. So I have a Th1, a prominent Th1 response. And he found some cells which are making IL-17. And the prominent Th1 response was actually not such a surprise for us, as already been shown by Markus Neurath, that actually the transcription factor Tbet is absolutely essential for T cells to be able to induce colitis. Yeah? So in this case, when he took T cells which are deficient for Tbet and used them in a T cell transfer colitis model, they actually were not inducing any colitis, yeah, as opposed to so it's looking at weight loss as opposed to the wild type T cells or the heterozygous T cells, which is showing an intermediate effect. And also, we looked at the histology, so the inflammation score, showing that the knockout Tbet knockout T cells actually were unable to induce any colitis. So when Jakob tried to reproduce his results, actually there we found kind of surprise. So first of all, he looked whether the cells are actually expressing TBAT. Yeah, so he saw that all the cells are expressing TBAT. But when he took TBAT knockout T cells, yeah, so here the control saying that we don't see any TBAT in these cells, actually he could see no difference between the wild type and the TBAT knockout T cell. Yeah, so very much in contrast to the work which has been published by Marcus Neuber. And also when we looked in the histopathology score, we saw that so the epithelial hyperplasia and the affected area were not significantly affected, but we saw some differences in the infiltration. Yeah? So the mucosal infiltration and the submucosal infiltration when we transferred TBAT deficient T cells. So then we were asking, so what is the difference between these two cells, and is it because of the microbiota? So what we did then, we took Charles River RAG1 mice, which came from Charles River, and then either he transplanted the microbiota from the DRFZ breeding facility or not, let the microbiota establish, and then induced the colitis by transferring either wild type or TBAT deficient T cells, and then waited whether they would develop colitis or not. And what we actually saw is that it seems to be very dependent upon the microbiota whether a T cell which is deficient for TBAT can actually induce colitis or not. So here's the mice which received the TBAT wild type or TBAT deficient T cells which had the microbiota of Charles River, so RAG mice which came directly from Charles River. So you see when you transfer the wild type T cells, these mice are very sick, yeah, a lot of infiltration, whereas when you transfer the T cell TBAT deficient T cells in the mice which are coming directly from Charles River, actually the intestine looks normal, yeah, and there's also no or very little histopathology. Whereas when we colonize the RAG mice beforehand with microbiota which is coming from our DRFZ animal facility, we see that actually here the TBAT deficient T cells are actually capable of inducing colitis. Yeah, and that we also see in the histopathology score, so there's some, it's a little bit reduced, but still they have massive inflammation. So apparently the microbiota composition can actually determine whether a certain functional repertoire of the T cell is required or not to induce colitis. Yeah? So in that case, being the functional repertoire which is controlled by TBAT. So we looked at the microbiota and saw that so if you compare before colitis induction, so in the healthy state, so this is the microbiota which is in the Charles River mice, and that is after colonization with the DRFZ microbiota. There's already, you see that there are differences in the microbiota, and this is also even more changed when the mice get colitis. So we did some prediction analysis to see, can we identify bacterial groups which are somehow positively or negatively associated with the phenotype that we saw. And we came up with a short list of bacteria which in the before colitis are associated with the susceptibility of TBAT knockout T cells to be able to induce colitis. So these are all associated with the susceptibility of the mice to TBAT deficient colitis. And there's some overlap, but also some new groups appearing when we look when the mice already have colitis. So what bacteria might be associated with the maintenance of colitis. So this is where we are at this point in this project, and of course now have to look, can we identify certain bacteria which are somehow making the mice susceptibility for, let's say, non-Th1 colitic inflammation, or protect them from this kind of colitis. So we have now identified all these different groups and saw also changes in the microbiota composition. And we thought, okay, is there maybe a different way than just 16S sequencing by how this was done? Whether we can identify somehow changes in the microbiota. So we looked whether we can actually use the flow cytometry to look at the heterogeneity and changes in the microbiota composition during colitis development. And we used the influx cell sorter to do this, and together with Susanne Müller from the Helmholtz Institute for Environmental Research in Leipzig, we actually tried to develop a technology by which we can look at the microbiota composition by flow. And Susanne Müller has already used flow cytometry to look at the microbiota composition in sewage, so in wastewater treatment plant, to look at the changes in microbiota composition during the processing of wastewater. And we adapted this technology for intestinal microbiota. So it's based on two parameters. So you look at scatter light and you look at the DNA content by Darby stain. And when you do this, here's such a cytometric profile. Yeah, so of course you get out the background and you spike in beads, so you can somehow standardize the measurement. And then what you see is this is the cytometric profile of the fecal microbiota of a healthy mouse, and this of a colitic mouse. Yeah, and you see very drastic differences between these two already by naked eye. And so we used the analysis technology was also developed in the lab of Susanne Müller, which they called CyBar, and that is basically a kind of binning. Yeah, so you make manual gates for all the populations that you can identify in such a dot plot. Yeah, and using all these gates, you make a kind of gating template. Yeah, and then you put this gating template on all the samples that you have measured. And this is then how it looks like. Then you can identify somehow regions and populations, and can compare changes in the frequency and distribution of these changes. And when you look at this statistically to find somehow are they more similar or very different, you can see that using this kind of gating strategy, you can actually clearly distinguish the colitic microbiota from the healthy microbiota. So but you can already see by naked eye, right? So when you do this. So we also looked, can we use actually this strategy to really identify a particular bacteria? So we wanted actually to see what is hiding behind these gates. So we took some of these gates which somehow define a very clear population and sorted these out, and then submitted them for 16S sequencing. And we were very positively surprised that we saw that these cytometrically defined gate actually constitute a very homogeneous group of bacteria. Yeah, so in all the gates that we sorted, at least 75% of the bacteria which were in the gate and were sorted out actually constitute a single taxon. Yeah, telling us that actually this kind of method can provide us sufficient resolution to really look at the heterogeneity of the microbiota. So we can also use this to look at changes over the course of time. Yeah, so we looked at regions which were either significantly expanded or significantly contracted during colitis, and followed these populations also over time. So sampled the mice repeatedly at several days after colitis induction, so meaning the transfer of the T cells. And here in the black lines is the frequency changes in these particular gates, so 44, 63, and 70. And here we see that these gates disappear. Yeah, so they disappear in the course of colitis. When other gates, when the frequency in some gates actually increases, so in 56, 71, and 77, and that these and there we correlated these changes in the microbiota, so in the frequency of these particular populations, to the appearance of clinical signs of colitis. So looking at weight loss and diarrhea score, and you can see that there is actually a time that happens at the same time. Yeah? So changes in the microbiota and the appearance of clinical symptoms. So there actually seems to be a causal relationship between these two. So if I summarize the first part, that actually show that the microbiota actually influences the susceptibility of the chronic inflammation, and that the pathogenesis of the chronic inflammation in our case is actually a combined result not only of genetics but also of the composition of the microbiota, and that you can use the microbiota cytometry not only for the monitoring of the microbiota composition during the course of disease, but that we could also use this to actually identify and directly isolate bacteria from heterogeneous mixture to do functional analysis without really having the need to cultivate the bacteria. So opening up us the possibility to actually look at bacteria which are so far not cultivated. So in the last part, I would like to give you an example of what we think are maybe a beneficial bacteria, because we think that it may strengthen the intestinal barrier and actually have an anti-inflammatory effect. And that was work done by a postdoc in my group, so Alexander Beller. And what he did is he compared the IgA levels in mice, which are at our breeding facility, and IgA levels in mice which are coming from Charles River. And he could actually see that there's a significant difference between in the IgA level from our breeding facility and in the Charles River facility. Again, pointing to kind of microbiota effect of what he sees. And this is actually the decreased amount of IgA is actually due to the decreased generation of IgA producing plasma cells. So here's the fecal IgA levels from a mouse from Charles River and a mouse from our breeding facility. And this corresponds to the number of plasma cells which he finds in the lamina propria, and also the number of IgA positive germinal center cells in the Pius patches, which we can hardly find in mice from our breeding facility. So it actually seems to be the generation of IgA expressing plasma cells which seems to be impaired. And this impairment can be reversed. Or this phenotype can be reversed when he puts the mice together, from a mouse from Charles River and a mouse from our breeding facility. So here's when he keeps the mice separate for fecal IgA, and here's the fecal IgA levels when he puts the mice together. Yeah, so it's an increase in the IgA levels from our facility, and this also corresponds then to the number of IgA positive plasma cells in the lamina propria. So really pointing to us that there's really a microbiota defect, and there must be something dominant in the microbiota of the IgA high Charles River mice, which then can induce the IgA high phenotype in the mice in our breeding facility. So we looked at what could be the reason for this increased IgA level, and as you know, TGF-beta, the cytokine TGF-beta, is actually the major driver or the only known switch factor for IgA. So we look for TGF-beta mRNA in the T follicular helper cells isolated from Pius patches. You could see that in the IgA high mice, there are also increased levels of TGF-beta mRNA in the T follicular helper cells as opposed to the IgA low phenotype. And this seemed to be very specific for TGF-beta, as we saw no differences in the interleukin-4 levels. And this TGF-beta expression levels could also be increased when he co-houses IgA low mice with IgA high mice. So really showing us that the microbiota actually controls the TGF-beta expression levels in the TFH in these mice. So we want to somehow move away from this kind of two different mice, and to really show that actually the microbiota. So what Alex then did, he took wild-type mice, from Charles River, and he treated them with young mice and treated them with antibiotics to deplete the intestinal microbiota. And then he transferred the fecal microbiota either of animals from the Charles River facility or from animals from our facility. So IgA high and IgA low microbiota, and then analyzed the mice 24 days later. And what you could see is that when he transfers the microbiota of the IgA high mice, those were able to induce, again, high levels of IgA, whereas when he took the IgA low microbiota, they were not able to induce high levels of IgA. And that also corresponded, again, with the number of IgA-positive plasma cells in the lamina propria and the number of IgA-positive germinal center B cells in the pious patches. So then we want to see what is actually the difference or the critical difference in the microbiota between IgA high and IgA low mice. So we again did 16S sequencing, and of course there are many differences in frequency of certain taxa. So we concentrate, to make the list a bit shorter, we actually concentrated on bacteria which are present in the IgA high mice and are not detectable in the IgA low mice. And when we used that filter, actually there were only 6 bacterial groups which popped up, which are shown here. And then we also compared them to the microbiota after co-housing. Yeah? So we again took another filter where we said, okay, these bacteria would have to be transferred after co-housing to be a candidate for an IgA induction. And there were actually only 2, the Anaplasma and the CytometryImmunity, which came up. But the Anaplasma got the higher score, so we started with this one. And the Anaplasma is actually not much known about the Anaplasma, except that they have no cell wall, yeah, and they are very small in size, so 0.2 to 0.5 micrometer. They are cholesterol deficient, so they may require other bacteria or host cells to be able to survive. And because of their small cell size, we thought we could maybe enrich them by filtration. Yeah, by using a 0.45 micrometer filter, and when we take the fecal microbiota and can filter them through a 0.45 micrometer filter, we can actually enrich this bacteria several long. Yeah, and deplete most of the bacteria. So we took the filtrate and sequenced it again, and could see that actually in our filtrate from the IgA high mice, only the Anaplasma more or less remain, and all the other ones, the candidate bacteria, in particular CytometryImmunity, are depleted from the filtrate. So then we want to check whether actually we can use the filtrate to induce IgA microbiota. So what we did is we took the microbiota of an IgA low mouse and supplemented it with the filtrate of an IgA high mouse. So again, we took wild-type mice, treated them with antibiotics. So this is when IgA levels when we don't reconstitute them with microbiota, so IgA levels stay low. When we take the microbiota from an IgA high mouse, we see that we can induce IgA levels in the mice. When we take the microbiota from an IgA low mouse, no IgA can be induced. But when we take the IgA low microbiota and supplement it with a 0.45 micrometer filtrate from an IgA high mouse, it looks exactly like when we take the complete IgA high filtrate. So indicating to us that actually the IgA inducing function has to be contained within this 0.45 micrometer filtrate. Also from these mice, we again did 16S sequencing to look at the microbiota. And here, again, looking at our 6 candidates, and we saw a different differential reconstitution of these 4 candidates. But when we compare it to the ability of induction of IgA, actually only 2
of the 3 mice which received the filtrate were actually reconstituted with aneuroplasma, and only those two only in these 2 mice IgA was induced. So there's actually a very good and very strong correlation between the presence of aneuroplasma, or the transfer of aneuroplasma, and the induction of IgA. And just as another thing that Alex did was to see whether about the TGF-beta inducing potential. So he turned to an in vitro system. We just took splenocytes from a wild-type mice and simulated the splenocytes using anti-CD3, CD28, and then added either the filtrate of the 0.45 micrometer filtrate containing aneuroplasma from an IgA high microbiota, or the 0.45 micrometer filtrate as a control from IgA low microbiota, cultivated them for 2 days, and then resimulated them with pyrimeonomycin, and then checked for TGF-beta induction. And here's the result. So when he has no bacteria, that's basically the baseline TGF-beta. And when he takes the 0.45 micrometer filtrate from IgA low mouse, also no TGF-beta induced, and the only sample where TGF-beta is induced is actually when he takes the 0.45 micrometer filtrate from an IgA high mouse and incubates them with splenocytes. Again, showing us that this TGF-beta inducing potential is actually contained within this 0.45 micrometer filtrate from the microbiota. So that actually, I could show that that aneuroplasma actually enhances the mucosal IgA expression, and that it does so by inducing TGF-beta expression in TFH cells in the Peyer's patches. And of course, as you know, that TGF-beta is also an important regulator of immune reactions, and IgA itself is also discussed to be protective against cholitogenic bacteria. So that we think that aneuroplasma, through the induction of TGF-beta and IgA, may actually have the potential to be an anti-inflammatory probiotic, and may be beneficial in circumstances of mucosal inflammation. So in the end, I'd like to acknowledge especially Andreas, who has been a very great mentor in this project, and has also have given a lot of input. Jakob, who has done the Tibet knockout studies and has started with the microbiota flow cytometry, and Alex, who has continued with the aneuroplasma IgA story. And of course, with a lot of work, and in particular Pavel, who has done all the bioinformatics and the analysis of the 16S sequencing. And then, of course, Andrea and Justus for the very nice collaboration. Susanne, without whom we could not have done the established the microbiota cytometry, and then the group of Peter Sigmund, with whom we now want to start analysis of IBD patient samples. And of course, in particular, the Rolf Schwiete Stiftung, who's very generously funding this project. And thank you for your attention.